Welcome to Ferment Radio, a podcast series on bacterial and social fermentation. Fermentation can incite social action, spark creativity, and bring surprising new tastes to our lives. My name is Aga Pokrywka, and I invite you to join us in a conversation on living interconnectivities, from macro to micro, from societal to cellular, and from global to personal. Dog's vomit, moon shit, demon droppings, snake poo. These are only a few names of certain types of slime mold, one of the most unusual and under-researched life forms on Earth. They can be found on every continent, but since they are not really useful or profitable, their presence has been largely overlooked by humans. Well, that was just before scientists discovered that even without a nervous system, slime molds can actually find their way through complicated mazes. This brings up a very interesting debate. What does it actually mean to be intelligent? Slime molds are neither slimy nor moldy, neither solid nor liquid. They escape all kinds of classifications. Could they be fungi because they produce spores, or perhaps animals because they move? Nowadays they are considered to be part of the Protista kingdom, but that might probably change too. Slime molds are like one big cell with plenty of nuclei, and they also have 720 different sex genders. In other words, they are at the same time one and many. Slime molds are really beautiful. They live in the forests, mostly in the tree stumps, dead logs, rotten wood, and leaf litter. They are bright and multicolored, but they grow in dark and damp places. These creatures are quite small and do things very, very slowly. So, in order to study them, one has to have the right equipment, like time-lapse cameras and magnifying glasses. But above all, you need an exceptional attention span and relentless dedication. Sarah Lloyd is this kind of a person. Sarah is a world-renowned naturalist, writer, and photographer. For many years, she has done groundbreaking research on slime molds, which she actually collects within close proximity of her own house in the eucalypt forest in northern Tasmania. Who is Sarah Lloyd, and what does she have to say about herself? By the way, this is the first Ferment Radio conversation that took place over a landline.
Hello?
Hello, here's Aga Pokrywka.
Hello? I can hardly hear you.
All right, that's so great to hear you. I know that you have an experience with identifying some even like a new species of slime molds. And I was wondering actually, how do you figure out, or how as a naturalist you understand, okay, this is slime mold actually what I am looking at?
Yes, well that's taken quite a while to learn. So after my interest in birds, from that I developed an interest in all sorts of other life forms. So I was doing a lot of surveys on birds, and I was asked, these were like citizen science surveys, monitoring birds in remnant vegetation in the bush, and I always had to correlate that with plant flowering times. So I got to know some people who were very into plants. I got to know all the names of all the plants around, and then also from that I became interested in fungi. But it was through my interest in fungi that I got to know about slime molds, because slime molds are often talked about as fungus-like organisms, even though actually when you really look into their life cycle closely, they're very different to fungi. Slime molds I suppose some of them superficially look like fungi, but most of the ones I collect are only about 2 millimetres high, so they are quite a lot smaller than most fungi that most people would see in the forest or anywhere. They are different in a lot of other respects. So fungi are actually more closely related to animals. So that the substance in fungi is mainly chitin, which is similar to our hair and our fingernails, whereas slime moulds are a completely different kingdom, completely unrelated to fungi, and actually more closely related to plants in that they're mainly made of cellulose. And what I'm interested with the slime moulds that I'm collecting is the fruiting body stage. So slime moulds have quite a complex life cycle. They have what's known as a plasmodial stage, which is the stage that moves about and feeds and is sometimes visible on logs and on the soil and on plants. That's often studied in the laboratory, and there's a lot of really interesting research about the plasmodial stage, but my focus is on the spore-bearing stage. So this is the fruiting structure that it produces the spores for reproduction. When I started my study of slime moulds, which was about 10 years ago, I knew nothing about them. So I was familiar with one or two species that are quite large, but I had no idea about anything that I was collecting. And so it took me quite a long time to figure out the ones that I was seeing, whether they were rare or whether they were common, and whether I could put a name to them. And at the time, and actually still, there are no guides for slime moulds in Tasmania. There's quite a bit of research and work being done in the Northern Hemisphere, and so all the books that I was using were from the Northern Hemisphere, and they didn't necessarily reflect what I was finding in Tasmania. So it has been quite a long learning process to figure out what species that I have found in the forest around home, and whether the species are new to science. So it was not something like I saw a slime mould in the field one day and said, oh, that's a new species. It's taken a lot of time to figure that out. And in fact, the 3 new species that I have described from my study site, I first misidentified them as Northern Hemisphere species, and it actually took a lot of work, a lot of DNA research from a researcher in the Ukraine to establish that they were in fact new species, new to science, and different to their counterparts in the Northern Hemisphere.
Could we say that there are some species of slime molds in the Southern Hemisphere which are not present in the Northern Hemisphere?
Yes, that seems to be the case. Mostly people think that they're always considered to be cosmopolitan, so the same species occur all around the world. The first species that I collected that turned out to be new to science was actually it's a very distinctive species, and I misidentified it based on photographs I'd seen of this species. The species I thought it was collected in Costa Rica, and it was known as a tropical species. And because Tasmania is a temperate climate, and definitely not tropical, I thought it was strange that it would turn up in Tasmania. And so I wrote an article about it, and as a result of that article, somebody chased it up. It ended up going once again to this researcher. He was working in the United States at the time, and once again based on DNA and some of the morphological characteristics of the slime mould, they decided that was a new species. So that was actually the first one I found that was a new species. And there has been another one, one of the first species I ever found. It was very distinctive. I thought, well, it's very distinctive. I must be able to identify this just looking at pictures in books. And once again, I sent it over to a researcher in Spain, and that one still hasn't been described. But we do know it's a new species, and it has also been found in New Zealand. So some of these species I'm finding seem to be also occurring in New Zealand. So there could be this Australasian group of slime molds that aren't found in the Northern Hemisphere.
You work and live in Tasmania, and
That's right, yes.
what I find extremely fascinating in what you do is that your research is basically like, what is it, like 2 kilometers, like a proximity of your house? Since you are observing something so small, like also how you keep sensitive on ever-changing like stages of life of slime molds, or maybe some new species coming out, because you are watching the same landscape, you walk the same paths.
That's right, and that's when I started, I realised that they're very unpredictable. So you never quite know where you're going to see them or when you're going to see them. And most of the studies on slime molds are done by people, usually mycologists who work in academia. They might plan a trip to a particular location to look at slime molds in the field, and they might be lucky to find something because they're very dependent on weather. So they often appear after a period of wet weather. And so I realised pretty early on that I was in a very unique situation where I could be here all the time and every day go out and find different slime molds. So after 10 years of searching for them, I've got a fairly good idea of where they're likely to appear and when they're likely to appear. So I've just made the most of that opportunity to keep studying them here. Slime molds can be quite conspicuous. They often appear overnight or in the early morning, and when they first appear they can be very brightly coloured. So they can be bright pink or white or yellow and quite conspicuous in the forest against the mossy logs, and it's quite dark in the forest because there's quite a thick canopy of eucalypts. So that's, I realised the advantage I had of living here, and I've kept watching for them. I must admit I'm not quite as conscientious in my searching now as I used to be because it does take a bit of a toll on the body. So searching under logs and crawling around on the ground is quite hard work. So you find them on the ground litter, lots of leaves on the ground, but also on logs. But there are quite a few vines in the forest here, and they'll go right up into the canopy, and they'll have slime molds right up in the canopy. So they're found anywhere there's organic material.
Do I understand well that when you were moving to northern Tasmania, you were not really planning to research on slime molds, right?
No, well, we've been here for 30 years, and my main focus when we first moved here was birds, so I did a lot of surveys of the birds, and then I gradually became interested in the plants and the fungi, and it was through that I just started noticing slime molds, and as I became more and more interested, I just searched more and more and kept finding them, and I think what it indicates is that they're much more common than people think. They're found anywhere there's organic material, and as you probably noticed on Instagram, a lot of people are finding slime molds now. I think possibly one thing that's contributed to this is the ability of people to take good photos with mobile phones, and also photographers are just, especially macro photographers, are really finding they're just such exquisite organisms to photograph, and they like that challenge.
And for you, what is something which the most fascinates you about slime molds? Like something which drags you to it? Would it be possible to even describe it this way?
I think what's really fascinating is, well, first of all, that they've probably been on the planet for 500 or 600 million years. So they believe that slime molds were originally marine, and they possibly colonised the land at about the same time as land plants. And they're playing this really important role in the ecosystem. They're breaking down dead organic material, and they're also feeding on bacteria. So one of their main functions in the ecosystem is to feed on bacteria and recycling nutrients. So in doing that, making soils richer for plant growth. And the other thing that's really fascinating about them, of course, is the plasmodial stage. And this is the moving, feeding stage of a slime mould that's like a giant amoeba. And except that, so amoeba is a single-cell organism with a nucleus and protoplasm. By the time several amoeba combine to form the plasmodium, it can have multiple nuclei, but it doesn't have any cell walls. And that's the thing that makes slime moulds different from any other organisms. This multinucleate, essentially cell-less body just enclosed in a cell membrane, and its ability to move. So they can move up to meters in a day, and they do this by a process called shuttle streaming, where they'll pulsate and they'll move forward for about 50 or 60 seconds. They'll stop for a while. They'll move back for about the same amount of time, 50 or 60 seconds, but they ultimately move forward, and as they're moving forward, they're feeding on bacteria and other microorganisms, the fungal hyphae, fungal spores, algae, and possibly other slime moulds. So they're absolutely fascinating from so many different aspects, and this ability to find their way through mazes and how researchers have used them to design transport systems. And people often send me these stories. They'll send me the latest story that's been reported on about this sort of research. And really, when you think about it, for organisms that have been on the planet for 600 million years, it's very easy to understand that they are very efficient at moving around and finding sources of nutrition.
Yeah, they are so hard to categorize, right? Like throughout the years, they were changing their place in the taxonomy, and they have different stages of life. They have this, I think, very unusual feature of having this many nucleus in one kind of a body, let's say.
They were originally, I think when Carl Linnaeus was devising his system of classification, they were thought to be fungi at that stage, and they went into the plant kingdom because fungi were thought to be plants. And then they went into the fungi kingdom, and then into the animal kingdom because of the moving feeding stage. And then they discovered this amoeboid stage, so the single cell stage. And they're now considered, they're part of the protozoa kingdom, I think, amoebozoa, protozoa. There's still a bit of taxonomic disagreement as to whether amoebozoa is a super kingdom or, whether it's a kingdom or not, or just a super group. But yes, they're very hard to categorize, and they have these different stages in their life cycle. So they have amoeboid stage, but the amoeba have the ability to form dormant structures called microcysts if conditions are unfavourable. And these dormant structures can live, I think they can live for a very long time in the soil. We don't really know because they're microscopic and indistinguishable from other amoeba, as well as being able to form a dormant structure at the amoeboid stage. They can also form dormant structures at the plasmodial stage. And so they have a whole lot of different stages in their life cycle. And the other thing about my research, I suppose, that makes it so interesting is that almost all the research about slime moulds is undergone in the laboratory, because some of the species do well in a herbarium or, an aquarium or just petri dishes, and they feed them on bacteria or rolled oats. And so a lot of the information that we read about slime moulds is based on very artificial conditions. And what I'm doing, of course, is studying them in the field and trying to get a sense of where they operate in the field, which is obviously very difficult to do because they have so many parts of their life cycle that are completely invisible to us.
I was also thinking about this, what we've been talking about just a couple minutes ago, about this difficulty of classifying slime molds is also like maybe the limitation of this kind of a classification itself. And then I was also thinking how, so we also mentioned about this ability of slime molds to figure out their path in the maze, and which was, I don't know, so popular some years ago, I guess, on the internet, all those articles. Yeah. And interestingly, this was the biggest surprise because they are kind of brainless and they look very basic. Maybe this also questioned, like, so what does it mean also the intelligence? Do you need to have a brain for that, and how your nervous system should be developed?
Yes, I mean, these are really unanswerable questions. On the plasmodial stage of the slime mold, and figuring out that they can find their way around to find food, which is something that you can understand. It's an organism that's been successful on the planet for 600 million years, but this notion that they can also pass on information to other plasmodia that have separated from them, and then it's something that we can't really understand because we don't know how they do it. But there is some sort of intelligence there. Or is it intelligent? That's interesting to ponder.
Because then we would need to question what is intelligence, right?
Yes. I suppose it's throughout the whole of the natural world. There are these senses that we don't completely understand. Plants grow to certain places and shun certain other places, you know. They grow towards the light, obviously, but I was listening to a program about bean plants and how they're attracted to particular plants, but they might grow away from other plants. Slime mold's just another example of one of these organisms that we don't fully understand, and how they transfer knowledge or throughout the organism. It's an unanswerable question at the moment, but interesting to ponder.
Is there some particular, maybe even personal learning which you gained through research on slime molds and through, hanging out with them out there?
Hanging out with them. I think how unpredictable they are, the unpredictability of slime molds. I was writing the preface for the third edition of my book. This was at the beginning of 2020, and I thought that I was quite familiar with when they appeared and where they appeared and what season they appeared in. And then last year turned out to be a completely different year. So they're totally unpredictable. There's so many things I've learnt that I didn't expect to learn through this research about slime molds, and still many questions that it'll take me years to actually find the answer to, I think.
So what's the rhythm of your work, actually? Is there some particular time of the day when it is easier to observe them, or...?
Yeah, well, as I said before, they do tend to appear. So fresh ones tend to appear overnight or in the early morning. I do tend to go for a walk every morning. So I just go for my usual walk through the forest every morning. And if I see something there, I will make a note of it or just mark it somehow, because they're often very conspicuous when they first appear, like they're brightly coloured, but they do, as their spores develop and the fruiting bodies mature, they become very inconspicuous, so I tend to mark it with a piece of tape. Sometimes I'll set up the camera and take what's essentially time-lapse photos of and showing how they develop, and other times I'll just go back and I'll collect the mature fruiting bodies. I've got them, and I'll bring them back to the house where I've got my microscope, and I'll spend a lot of time trying to figure out their identification. And so I have got a herbarium, a personal herbarium with about 2,000 collections in it now, and I also send a lot of samples to the herbarium, like the National Herbarium. If overseas researchers want to use any of my material for research, they can access anything from the herbarium, the National Herbarium. So, yeah, it's quite a time-consuming process. I do collect, if I collect species in the field, I bring them back. They dry very quickly. They air dry in an hour or so. then I mount them on card, and they do last indefinitely. So if they're stored properly, they'll last for a lifetime. And in that respect, they're very different from fungi. So if you collect fungi, you've got to describe them straight away because they shrink. With slime molds, those features will be there indefinitely.
Do you have any like a favorite or special, well, besides the one you discovered, kinds of slime molds you would like to tell us about?
Yeah, there's some really beautiful ones. I think the ones that probably are most popular are the iridescent ones. So the membrane that surrounds the spore mass is called the peridium, and a lot of the species called Lamproderma have this beautiful iridescent peridium. So you see all these beautiful blues and greens and pinks and iridescent colours. And I suppose the other thing that really fascinates me about them is that once you get into looking at the microscopics of these structures, they're just exquisite. In fact, some of the tiny little fruiting bodies, if you look at them with all their spores blown out, just the bare structure, and they're very like miniature trees. So I look at these giant trees in the forest around home, and they're very similar to these, 1 to 2 millimetre high slime molds that I'm collecting in the forest. It's a sort of interesting comparison, this huge thing to this tiny organism with similar structure in their branches and stalks, trunks.
Maybe it's a little bit unfair how slime molds are named, right? Because neither slime or mold is really like, it doesn't sound very attractive, right? But what you describe doesn't really fit this name.
I know, it's very unfortunate, and it was named, I think they got the name slime mold in about 1833 when a German mycologist gave them the name myxomycete, which translates to slime fungus, actually. So that refers to the plasmodial stage of the life cycle. That's where they have multiple nuclei and they're just one large cell. It's actually a very interesting structure, so it's full of food vacuoles, but it is covered in what's called a slime sheath, and that's like a slimy layer, and that stops the organism from drying out. Unfortunately, has given the whole group this unattractive name, when in fact they're very beautiful organisms.
Next time when I go to the nearby forest and I would like to look for slime molds, could you give me some hints how I should look for them?
Well, I always take a very strong light, torch, hand lens, torch or hand lens or flashlight, and I always have a 10 times little magnifying glass or loop. I generally shine the light on, and the places I look most would be if there are big logs, and where I live in Tasmania, the forest had some logs taken out, but there are big logs that were left on the forest floor, and they're now covered in mosses and lichens and liverworts, and at certain times of the year they can be very productive. And also I look on the leaf litter and even standing dead trees, just anywhere. But it takes a bit of practice to figure out whether what you're looking at is a slime mold. Sometimes very small fungi can resemble slime molds, so it does take a bit of practice. And they generally appear a couple of days after rain. That's when I usually go out. So if it's been raining through the winter, it can be very productive. So it's summer in Tasmania at the moment. There isn't much happening, but I have found a few in the last. So we have rain periodically through the summer, or certainly this year. Firewood piles and any woody material that's nice and wet, you're likely to find slime molds.
Did you ever find like a species of a slime mold which you were not able to find again?
Yes. Well, I have found one or two species only once or twice. So some species are really, really common, and I find them every year. Some species I find some years, and I don't find them again for another couple of years. And there are some species I've only ever found once or twice. But it's taken me a while to figure out which ones are the rare ones. In fact, one of the first species I ever found was a beautiful iridescent one, and I was sending some samples to a friend at the time, and he couldn't find any reference to it, and he eventually tracked it down, and it was a species found in Japan and was supposed to be quite rare, and it's actually turned out to be very common here. It reflects not just how unpredictable they are, but just how few people are studying slime molds. So I'm the only person in Tasmania who's really studying them regularly.
Why the research on slime molds is so unpopular?
I think I have read that they're the least studied of all the microorganisms. And I think the reason is because they're unpredictable. So if a mycologist who studied them in the field, because it's so hard to anticipate because it depends on the rain, then it's hard to plan a trip in advance to study slime molds. One of the main ways that they're studied is that people go into the field, they'll collect any fruiting bodies that happen to be in the field at the time, but mainly what they do is collect substrate. So they'll collect bark off living trees, they'll collect leaf litter, woody substrate, and they'll take this back to the laboratory and they will culture it in petri dishes. But they have to be checked very regularly to see if any slime mold fruiting bodies are appearing. And it's a very time-consuming process. And actually, I just said that they're the least studied of all the microorganisms. That's at the fruiting body stage, which is the stage I'm studying. At the plasmodial stage, they're one of the most commonly studied organisms because people can keep them in the laboratory and feed them and watch how they move and behave.
That's what's so fascinating about the work of a naturalist, to be able to observe and study slime molds in their own environment as what they really are, a unique and unpredictable species. Something you might be now wondering is why Ferment Radio made an episode on slime molds. Well, they feed on bacteria, have been overlooked for a long time, and they are very hard to classify. But most importantly, because of their own nature, they are capable of questioning human main paradigms. And this is what Ferment Radio is about. If you would like to know more about the show, listen to this episode again, or find previous episodes, please go to fermentradio.com. You can subscribe on Spotify, Apple Podcasts, and more. I'm always looking forward to hearing from you at hello@fermentradio.com. Ferment Radio is brought to you by Culture of Cultures and is produced by Super Eclectic. Thank you for listening. Keep fermenting and stay tuned for the next episode of Ferment Radio.